So, hey learners, welcome and welcome back. I hope you are doing all good. And we were left with the last topic called metabolism. So, we'll see the topics to be covered in this chapter are metabolism and its type called primary and secondary metabolites. Then we'll have concept of metabolism, which is the dynamic state and their body constituents. We will see and a metabolic basis for living and the living state. This lecture will hardly take your 5 to 10 minutes. So, let's begin by the basic definition of metabolism. Set of all chemical reactions occurring in living organism called as metabolism. And due to these biomolecules, which you already know, once they take part in the metabolism automatically becomes essential for the survival of all living organism. Then we have metabolites. They are the small molecules and the intermediate product of metabolism. It means they take part in the process of metabolism. And other than that, biomolecules also take part in the process of metabolism. So biomolecules are also known as metabolites. And once these metabolites are converted into each other in a series of multi-step chemical reactions linked with each other, during the process of metabolism, they form the metabolic pathway. And remember, majority of these metabolic reactions are always linked with some other metabolic reactions occurring in our body, where each of these steps is catalyzed by a specific enzyme in between. Now we have certain features which affect metabolic pathway. So by a simple analogy of car, you will understand what I am saying here. As you need to fuel your car for the proper running of the engine, similarly you need to fuel your body not by the petrol but by the food you eat for the proper metabolism. So let's start by the features. The metabolic pathway are interconnected or linked with each other. It means the product of one pathway can become the substrate for the another pathway. Suppose these metabolic pathway are similar to the automobile traffic in a city. As you can see in the image, a road full of traffic where cars moving one behind the another. Similarly, metabolites like glucose, fatty acid, amino acids move behind each other of metabolic pathways. Then metabolic pathway are irreversible as they proceed in only one direction because they involve a large energy change. Now these pathways are either linear or circular. Now imagine these linear pathway like a one-way road trip where car A followed by car B in the same direction, and once the journey ends the car doesn't returns. It means the metabolic pathway will not repeat in a similar direction, where every stop like the fuel station acts like an enzyme which helps the car to reach the destination. For the example, glycolysis, where the glucose converts into pyruvate and then stops there. Whereas car running on a circular race track, after completing one lap they return to the starting line and can go round and round again, where checkpoint act as an enzyme that help the car complete the loop, and the cycle continues as long as the fuel substrate is supplied to the car. For example, we have Krebs cycle in our cell which converts from one form to another repetitively in a single direction. Then there is one more branched metabolic pathway where the pathway crisscross each other, that to split into two or more directions like a road with multiple exits. Think of a highway full of traffic junctions. Now the road you want to take up depends on few factors like the traffic signal. If the traffic is dense, you will choose some another path. If the traffic is low, you will go through that way. Similarly, in metabolism, enzyme control the flow of reaction just like the traffic signal controls the car at a road junction by the help of traffic lights yellow, green, yellow and all. When to go green, yellow to slow down the reaction and red to stop the reaction. It also depends on the fuel ability. In a car, if you don't have petrol, the car cannot run no matter how good the engine is. Similarly, substrate levels, which is equal to the number of car enters on the road. If there are enough car, which means the high substrate, which leads to the fast traffic flow because of the high reaction rate, and if there is few cars, which means low substrate, leads to the slow traffic flow because of the slow reaction rate. These analogies are provided for your conceptual understanding, not to confuse any one of you. So repeating it again, if there is no car on the road means low traffic. Similarly, no substrate, no enzyme going to work, so no product will form. And Fully fueled and many cars on the road means dense traffic, road is busy and engine runs very well. Hence the high energy and substrate leads to the fast metabolism and energy production. And then we have destination need. See, it's quite basic. In our body, the cell decide which product is needed to form according to the situation. Similarly, the driver decide where to go depending on the purpose. Different needs lead to the different metabolic pathways. Now imagine all road signals and vehicles work properly. Traffic flows smoothly without any jam, just like metabolism flows smoothly in a human body without a single report or mishap for maintaining the healthy condition in human body. But what if any signal breaks, accident or fuel shortage occur which can lead to traffic jam and can turn into a big city chaos over there? Similarly, if any single mistake is reported in the metabolic pathway like enzyme failure, missing substrate, which can alter the complete human body condition and can lead to many metabolic disorders. Now the another feature of these metabolic reaction is that every chemical reaction is a catalyzed reaction, and the catalyst which increase the rate of given metabolic conversions are also called proteins, and these proteins with the catalytic power are named after the enzymes. Just like cars which required good fuel service and smooth roads to reach their destination efficiently by shortcuts. Similarly, enzyme makes sure in the body reaction happen faster, properly with less energy requirement. For example, glucose converts into pyruvate, which is catalyzed by the enzyme, and even carbon dioxide dissolving in water is a physical process but a catalyzed reaction in the living system. In short, what I'm trying to say is there is no uncatalyzed metabolic convergence in living system. Then we have the fifth point which says the metabolic flow is called the dynamic state of body constituent. So we'll see this point in the living state topic. So let's move by the another slide. Now you may ask why metabolic flow is called the dynamic state of body constituents, because the molecules in our body are constantly being broken down, rebuilt and recycled. They are not static or fixed. So the dynamic state is like the water in a flowing river. When there were some molecules are always in a continuous movement even when things look balanced. So as you can see on the screen, we have cars as metabolites, where we'll see how fast metabolite A converts into B is called the metabolic flux or the rate of metabolic conversion, where the arrow indicates the direction of the flow. So the number of cars on the road may stay same, but they are always moving or entering, exiting. Similar just like to the metabolites they are always being transformed even when they are at the rest. This metabolic flux actually represents the speed. Flux is like the traffic flow of metabolites, where the higher flux means the faster movement of the molecule, where traffic moves faster, and the lower flux means the slower movement of the molecules, where the traffic moves slower. So what we have learned till now is that living organism, either it's a protozoa or just even a cell, any plant or animal, they are all contain of 1,000 organic compounds in them, and these compounds are the biomolecules which is present in them at a certain concentration. So one of the greatest discoveries ever made was this observation that all these biomolecules have a turnover number. So in the process of metabolism, biomolecules get converted, or you can say turned over, from one form to another form of biomolecule, and this keeps on happening. So the constant formation or change from one biomolecule into some other biomolecules. This breaking and making is throughout the whole complete chemical reaction constantly occurring in living organism. As the biomolecules are present in certain concentration, which is a mole upon cell or moles upon liter and etc. Similarly, the turnover number is expressed as amount upon time. They are usually amount upon time. For the example, conversion of any XYZ milligram amount of compound per day or mole per hour. There is no fixed unit of amount and time. The amount varies as per the compound, and the time goes from second to hour and even up to days as per the conversion of their molecules. Now we will see what is metabolic transformation. So basically, when each of these metabolic reactions result in the transformation of biomolecules called metabolic transformation. We have few examples, we will see them. So the removal of carboxylic group adds CO2 from amino acid mix, and amino acid turns into amine by the help of enzyme called decarboxylation. Then we remove amino group as NH3 from the nucleotide base. It undergoes deamination to form some other different nucleotide base. Then the third one, where hydrolysis of glycosidic bond in a disaccharide to form other monosaccharide units, where you can see the carbohydrate, which is a complex food, get digested into our body in the form of simple food called glucose. Similarly, you can understand this. Now don't be confused with the metabolic turnover number and enzymatic turnover number. They both are the different thing with the different perspectives. So the enzymatic turnover number measures the enzyme efficiency. You already know the number of substrate molecules converted into the product by the help of one enzyme in per second of time. But the metabolic turnover number actually measures the overall metabolic activity of a cell tissue in an organism. Okay, by the rate at which metabolic compound is synthesized or break down into the metabolic pathway, such as the synthesis and degradation of protein, lipid, ATP in the body, and often expressed as the mole per unit time. You already know the example of enzymatic turnover, where a single enzyme carbonic anhydrase converted 6 lakhs of carbonic acid within a second, whereas in metabolic turnover number, where total body ATP turnover is 60 to 70 kgs per day. That's the total amount of ATP used in our body every day, which is roughly equals to the body weight of the individual. So now it became easy to conclude that one is about a single enzyme activity and other is about the overall renewal rate of cellular components. Then we have two types of metabolic pathway. The first one is anabolic pathway, also called as biosynthetic pathway, and then second one is catabolic pathway, which is also called as degradation pathway. So we will see why it's called as anabolic or biosynthetic pathway. It actually a building up process where small and simple molecules combines together to form larger and more complex molecule by the use of energy, where we have this example of photosynthetic reaction from our very previous classes, where small molecules like carbon dioxide and water combines together in the presence of chlorophyll and light to form complex molecules like glucose and oxygen. Then we have catabolic pathway, which is just opposite to the anabolic pathway, where complex molecules combines together to form simple and small molecules by releasing energy. We again have this example, very basic example of respiration from our 7th or 8th class maybe. So when the complex molecule like glucose and oxygen breaks in the presence of anaerobic respiration into simple molecules like carbon dioxide and water to release energy in the ATP. Then we have this example where glucose is degraded to lactic acid in our skeletal muscle, where the energy is liberated. So this complete metabolic pathway occurs in 10 metabolic steps of glycolysis, where glucose is converted into the pyruvate, and when oxygen is absent in the anaerobic conditions, that pyruvate is further reduced into the lactic acid. We will see all these 10 steps detailed in chapter 14. So now we have this topic called ATP bond energy. As we saw in the last slide, whatever the amount of energy liberated during the during the degradation of glucose into lactic acid, living organism actually traps that energy which liberated during that degradation and store it in the form of chemical bonds, and then whenever they needed that energy, this bond energy is utilized for the biosynthetic, osmotic and mechanical work that we perform. Now we will see why this ATP bond energy is the most important form of energy currency in the living system through this diagram. Now look at this diagram. We know that ATP is the energy currency of our cell, but you ever wondered that why ATP is so high, rich in energy? Because it has three phosphate groups in it, which is called alpha phosphate, beta phosphate and gamma phosphate bonds, which is interlinked with each other with a very high energy phosphoanhydride bond, where this beta phosphate share its bond with adenosine. You already know what is it from the last lecture. It's a compound of adenine base and ribose sugar. So ATP stores all its energy in these phosphate bonds, and once you break these bonds by the process of hydrolysis, the ATP breaks down into ADP, which is adenosine diphosphate, and inorganic phosphate, and all the stored energy will release out and will be used in the metabolic activity. And the energy released is about 7.3 kilocalorie per mole as written under the standard condition, and the cycle of ATP to ADP, ADP to ATP continues throughout the cell. And the topics like how do living organisms drive their energy, what strategies actually they have evolved, and how do they store this energy and in what form, and how do they convert this energy into work. And you will study all these concepts later on in your bioenergetic branch of higher classes. So we'll see the living state. So basically there are thousands of chemical compounds which are called metabolites or biomolecules in the living organisms, which are present at a specific concentration, and even each of the biomolecules have a different concentration from each other. For the example, we have the blood concentration of glucose in a normal healthy individual, which is a 4.5 to 5 millimole per liter, and similarly we have the blood concentration of hormone in a normal healthy individual, which is a nanogram per ml. So the constant flow of matter and energy is maintained through the continuous biochemical reactions. It could be anything like protein, carbohydrate, lipid, nucleic acid, but let's take this example. As we saw, the blood concentration of glucose is 5 millimole per liter. It will remain the same throughout the complete biochemical reaction to maintain the dynamic steady state. If the value goes above and below the 4.5 or 5 ml per liter, it will create a problem. We will see what kind of problem in the upcoming slide. And the most important fact of biological system is that all living organisms exist in a steady state, dynamic steady state, which is characterized by the concentration of each of these biomolecules, and these biomolecules are always in a metabolic flux. We will see what is metabolic flux in the next slide, and we will also see why these steady states is a non-equilibrium state. So let's begin. So the number of molecule, or you can say turnover of molecules passing through a metabolic cycle called the metabolic flux. So basically it tells how fast substrates are converted from one form to the another in a series of metabolic reaction. You can take the example like total number of glucose molecules get converted to the pyruvate in the process of glycolysis so that they can form ATP and NADP. So that number of glucose molecules actually defines the metabolic flux in that particular organism. I hope this example is helpful for you. For example, suppose when you go to gym, you need more energy to lift all those heavy weights. So the metabolic flux increases through the glycolysis by breaking down more glucose molecules. And if you already have sufficient amount of energy, then the metabolic flux automatically slows down. And the metabolic flux actually matters a lot to our body. It allows the body to respond to changes like hunger, exercise, infections, or any other thing. And without it, we wouldn't be repair, damage, grow, or adapt even. And it also helps to maintain the internal balance in our body. Now I have a question for you all, that why biomolecules are always in a metabolic flux. You can pause the screen and can answer me. So according to physics, we already know that the system at equilibrium cannot perform work, and that is the reason why living state is in a non-equilibrium steady state to be able to perform, so that it can be able to perform work. And that is the reason why biomolecules are always in a metabolic flux. And also there is a point that after death, only after the death, for the next 10 to 15 minutes straight, equilibrium will maintain, and the body work done by the system is zero. And this non equilibrium steady state can be only achieved by the energy input. And there's one line written in your NCERT, that the metabolism provides a mechanism for the production of energy. Hence the living state and the metabolism both are synonymous to each other. Without metabolism, there can't be a living state. Now there is a term called BMR, which is basal metabolic rate. This topic is off from your NCERT, but I must say that you may have heard about this topic, so it becomes quite familiar for you all. So let's discuss. Have a look. Okay, so BMR refers to the rate at which the body burns energy or calorie to carry out the basic life functions like the breathing, digestion, and the heartbeat. So let's suppose whenever we say this X person has high metabolism and this any Y person has low metabolism, what does this terms actually means? So we'll see that first. Then we have two types of BMR. The one is high BMR, or also called high metabolism, where the person uses up more energy even at the rest condition when body is doing nothing. And the person feels more energetic in the gym, and may can find it easier to stay slim, and can also burn a lot of calories faster than the low BMR. And the low BMR, or also called low metabolism, where the person uses less energy and slowly, and can gain weight more easily than the high BMR, and feels on low on energy, and also they requires less food to maintain their body weight. So let's get back to the example. When someone says this X person has high metabolism, it means that their body burns calories much faster than any average person, and so that they may can eat more but still stay slim. So whenever someone says this Y person has low metabolism, this means that their body burns calories much slower than any average person. So whenever they eat enough, they get fat. So because of that, normal meal may provide more energy than needed for them, and the excess calories are not burned. They are going to stored and used as the fat. So they really need to maintain this properly. Now we will see why metabolism varies from people to people. So these are the few factors, we'll see them. First one is genetics. So see, some people are naturally fast calorie burners, or some are slow calorie burners. So it depends on their genetics. The other second one is muscle mass, because muscle mass actually leads to the high metabolism when muscle uses energy at when the body is at rest condition. Then the third one is age. Age plays a vital role, when metabolism usually slows down when the person starts getting elder with the age. Then we have gender. Gender is quite specific. Men often have higher metabolism as compared to the women. Then we have hormones. There are few hormones which plays role in the effect of metabolic rate, like thyroid hormone, insulins, and etc. Then we have activity level. More physical activity increase metabolic rate, we already know that. Then we have diet. See, diet matters a lot on many bases, like what kind of food type you are taking, and on what frequency you are taking your meals in a day, and the calories intake, it matters a lot. Then we have health conditions like hypothyroidism, when the thyroid gland starts producing too much hormones which lead to the high metabolism. And then we have hypothyroidism, when the thyroid gland produce little hormones which can actually slows down the metabolism. Now we have types of metabolites. This topic was supposed to do earlier, but anyways we'll cover it now. So the difference between primary and secondary metabolites. Primary means the main prior one, organic compounds, where they have the identifiable functions, so they directly involve in the main physiological process like growth, development and reproduction of the organisms. And then we have secondary metabolites. They are the derivatives of the primary metabolite, and they have the non-identifiable functions, and even they are not directly involved in any growth or any physiological functions. They also take part in the ecological functions. And these primary metabolites are same in all the plant species because they perform the basic life functions, as I already said: respiration, photosynthesis, protein synthesis, DNA replication. They are the common to all the plants. Okay, so all the plants make the same primary metabolite because they all do the same basic life activities. Then we have secondary metabolites, which are unique to the each plant species because they are not essential for the survival of the organism. See, different plant faces different challenges in their environment to survive, and each plant evolve in a unique way. So they use some chemical weapons or attractants, such as we'll see alkaloids, essential oils, we'll see in the slide. So they are unique to each plant because these compounds are not made by all the plants, only they are made by only those plants that need them. Now we are at the last point, which is the primary metabolites are produced in large amount and extraction is competitively easy as compared to the secondary metabolites. We'll see what this term means. So they are essential for the basic life process, and the plant need them constituently so that they produce in large quantity, and that is why their extractions become competitively easy. By their standard methods like crushing, boiling or solvent, extractions can easily separate them. Then we have secondary metabolites, which are produced in small quantity, and even extraction is competitively tough as they are made in the small quantity because they are not required for the daily survival of the plant, and even they produce when they are under stress or any kind of attack at certain stage only. And even their production is highly regulated and often costly for the plants, so the plants make only small or necessarily amount which they need, and that makes their extraction difficult because they are often stored in some special structures called vacuole, and they have the complex solvent extraction method and also advanced purification methods used in it, and many of them are unstable, okay, or degraded easily during the extraction. So we are already done by the examples of primary metabolites like glucose, carbohydrate, amino acid, lipids. Now we only left with the examples of secondary metabolites. We'll see them. And there's one more difference, that the primary metabolites, they are not poisonous, but secondary metabolites in them, like some of these compounds are poisonous, absolute poisonous. So let's begin by the next slide. Now we have this table 9.3 where we'll discuss some important secondary metabolites. This table is only from your NCERT and also becomes an important part of your syllabus. So the secondary metabolite is classified into 7 main classes. So let's begin by each of them. So first we have pigments like carotenoids and anthocyanin. So on the basis of solubility, carotenoid is lipid soluble and anthocyanin is water soluble. If you remember the last session where we saw lipids, so the carotenoids actually belongs to the terpenoid class of lipids. Not just carotenoids but chlorophyll also. Now as we already know that photosynthesis is a characteristic feature of chlorophyll pigment but not of carotenoids. But still chlorophyll and carotenoids together, they can perform photosynthesis, but the main function of carotenoid is to attract the pollinators. And their color range varies from yellow, orange to red. Their examples are beta-carotene, which is present in the carrot, and xanthophyll, which is a yellow pigment present in fruits like papaya, orange, mango. And then we have leucopene, which is a red colored pigment responsible for the color of tomato and chilies. Then we have water-soluble pigment anthocyanin, which is present in the vacuoles of the leaf, and their function is to attract the pollinators by their bright color. Their color range varies from red, purple to blue. Red color is for the rose petals, cherries and apples. Purple is for the grape and cabbages. Then the blue is for blueberries. Then we have alkunite. It's a group of nitrogen containing secondary metabolites which are produced by the plants. Then we have two of the most well known examples of alkaloids, which are morphine and codeine. You may have heard about their names before. So they both are derived from the opium plant. So first you need the license from the government of India to grow these opium poppy plant, which is also called as afim in Hindi. Once you grow these poppy plants and they get mature, you can make a shallow cut on the surface of their pod, as you can see on the image. Then this white milkish fluids comes out, which is called latex. Then you have to wait for the overnight while these latex turns brown and sticky when exposed to the air. Then you can collect all these next day early morning by your hand. By doing all this, you will get the dried latex, which is called the raw opium. This opium contains about 10 to 15% morphine in it and some 2 to 3% codeine. See, it has many useful benefits. If you use it in a low dose, it can work as a very good, helpful medicine. But it is also sold by the narcotics illegally in the black market. That is the reason why the production of opium is banned in India. The supply of high dose afim is also harmful for the drug addicts. Then we have another secondary metabolite, which is called terpenoids. So they are the isoprenes with a large group of natural organic compounds made from the repeating unit of isoprene units. So these are found in mostly in plants and serve the various functions such as aroma, flavor. So on the basis of number of isoprene units, we have monoterpenes and diterpenes. Monoterpenes has 2 isoprene units. And because of its aroma, it is found in essential oil, perfume, flavoring agent. As you can see on the screen, we have linalool, limonene, bisabol. These are the flavoring agents which gives flavor to the limonene, gives citrus scent to the orange and lemon. Minolin gives the minty compound in the peppermint and also to the mint leaves, and pinene gives aroma to the pine tree. Whereas myrcene is found in bay leaf, lemongrass, used for its aroma in perfume and flavoring agents. Then we have bisabol, used in skincare and cosmetics. And caryophyllene found in black pepper clove, which is used as a spicy aroma. Then we have diterpenes, which is made up of 4 isoprene units, which is responsible for the plant hormone and pigments. Vital find in the chlorophyll of all the plants. Taxol is extracted from the bark of yew tree, used for anti-cancer drugs. Gibberellins is found in all the plants, especially in the germinating seeds, which is a plant hormone, promotes growth and germination. Then we have essential oil, which is highly aromatic, extracted from the plants. These oils are rich in secondary metabolites. With the example lemongrass oil, used for the aroma therapy, it has very calming and refreshing vibes. And lemongrass tea is also quite familiar among us. Then we have another secondary metabolite, which is toxins. Plant produce these toxins just to protect themselves from insects and other microbes. These toxins are not used in plant growth or any development, but they are just vital for their survival, for their defense from the harmful animals and insects that they might not eat their seed, and it has no medical use due to its extreme toxicity in it. Then we have the example rosary pea. These are the bright red colored seeds with a black dot on it. In the ancient time, these seeds were used as the standard weight, which is called ratti in Hindi. Its weight is about 0.91 milligram and its weight is fixed. Every seed has this similar weight and it is fixed. That is the reason why these seeds were used as a traditional measuring unit to measure gold and any other expensive pearls. And these were anciently also used to make the jewelry and prayer beads because of their uniform size and weight, where 1 gram is equal to 5 pieces of ratti seeds. But other than its beauty, it is highly poisonous in nature because it has aberrant content in it. If you chew a single seed, it can shut down all your organs within an hour. It is deadliest harmful. And if chewed, you remember there is no known antidote for it, and it is 100 times more deadly than ricin. In short, it would not be wrong to say that it is a paradox of beauty and danger at the same time. So next time once you see this, just stay away from all these dangerous plants. Then we have ricin, which is again a toxic found in the Ricinus communis plant. Their seeds are called castor and arandi in Hindi. See, the entire plant is toxic, especially the seeds. But once you heat the seed, you can extract castor oil out of it. The oil is free from ricin because ricin is water soluble and do not mix with the oil during proper extraction. This oil can be used as cosmetics in skin care and hair care, but it can also be consumed as a vegetable oil, but only the purified refined castor oil. Even it was used in the past as a biochemical weapon. So protect these seeds from being eaten in even a tiny amount can cause death. It is extremely dangerous. Lectins is a compound produced by plants and organisms. They are not involved in growth, reproduction, development, but they help mechanism against herbivorous insects and other pathogens. And chemically, they are carbohydrate binding proteins, especially binds to the sugar present on the surface of cell, which is glycoprotein and glycolipids. Unlike other enzymes, lectins do not modify the sugars, but they bind, they just recognize and attach to them. The best example of lectins is concavline A. It binds to the carbohydrate on the surface of insect gut cell wall just to inhibit their bacterial and fungal growth and also to disturb their digestion and nutritional absorption. One major category of secondary metabolite include these medical compounds or drugs that can be extracted and used in the pharmaceutical companies. Plants produces a wide variety of these for their own protection and humans use them in medicines. One such plant is Vinca rosea, commonly called Sadabahar in Hindi. I'm sure we must have seen these flowers in our home and our locality. These flowers petals actually contain the drug called vinblastine. It's an anti-cancer drug used in chemotherapy. It can affect different type of tumors by preventing cancer cells from multiplying. Then we have our another drug compound which is curcumin. It's a yellow colored bioactive compound found in the turmeric plants and it is widely recognized for its medical purpose and can be used to treat many diseases. Curcumin gives the turmeric root yellow color and medicinal values to protect as a protective agent and against these microbial infections. It has been an important part of our tradition. We are using it for centuries in skincare, cooking, and medicines. But nowadays, our haldi wala doodh has become extremely popular drink in the Western countries. They call it as turmeric latte. And as you already know, people's filming this haldi oil water trend. Please remember, haldi is not just a color for camera. So don't waste it just for a visual or satisfying video effect. Then we have polymeric substances. We already know what is it from the last lecture of a polysaccharide carbohydrates. So these are basically the substances with the large molecule made up of repeating unit of monomers. Only some of these polymers are considered as secondary metabolite, especially when they are not essential for basic survival but serves as a specific ecological and protective functions. Depending on their role and origin, only some of the high molecular weight compounds like natural rubber, gums and cellulose can function as secondary metabolites. So let's see them. Rubber is a polyterpenoid polymer extracted from the latex of the rubber plant. A diagonal cut is made on the bark of the tree. Make sure that it is not too deep to avoid the damage. The cut is made in the early morning when the internal temperature of the tree is highest and the latex slowly flows out like water from a tap, and that is the reason why this process is called tapping. As you can see on the image how latex is being collected, it is slightly sticky in nature and it is roughly 55% water and 40% rubber content in it. The collection takes 2 to 4 hours per tapping and the latex naturally begins to coagulate if it is left in that bowl or coconut shell. As you can see, they are preserved with ammonia for the industrial use and mixed with chemicals, a lot of chemicals to make products like tires, balloons, gloves, footwear, conveyor belts and many such things. Then you can after that you can repeat the process, but remember trees are tapped only on alternate days or in a rotation to allow so that the tree can heal itself, and the cuts are moved slightly upwards or downwards over the time to extend its productive life. Don't forget, over tapping harms the tree and reduces the latex content. Latex production depends on the tree age, weather, soil, and the tapping skills. So take care of it. Then we have the another natural polysaccharide, which is gum. Gum often have branch structure which help them to trap water and form gels. Plant gums are extracted from the bark of acacia tree, babool, which gives Arabic gum. Why it is called Arabic? Because the acacia tree grows mainly in Africa, and the gums were first introduced by the Western world by the Arabic merchants. Then we also have Stirculia tree, which gives Kraya gum, which is called Gond Katira, and the previous Arabic gum were called Gond. Now the gum is extracted from certain trees through a natural or artificial process called tapping, similar to how rubber latex is collected. See, a natural process is when a tree gets injured, it naturally produces the gum to heal itself. Now we'll see it by tapping. First, small cuts are made in the bark of tree using a very sharp tool. This stimulates the tree to release a sticky sap called gum. As then the natural gum starts creating out the cut and starts to get hardened when exposed to the air. As you can see in the image, after a few days the dried gum carefully collected by hands. You can repeat this process every week during the dry season. And as I said, gum is made up of complex polysaccharide which makes it possible to soluble in water and useful as a thickener or emulsifier in food and pharmaceutical industries. It is sticky in nature and water soluble. Once you add a tiny amount of gum in water, it gets swells out. It is also used in food industry as a stabilizer in ice cream, sauces and cosmetics, and also as adhesive due to their sticky nature. The brown color gum, as you can see on the left side of the screen, is used during winters to make laddu, and the white color gum is used in summers to make refreshing drinks out of it. No doubt these gums comes with very useful benefits and also very popular in our Indian households. Then we have cellulose. We have already learned cellulose. We have a brief idea about it. Then we'll still see. See, cellulose is extracted mainly from plant biomass such as wood, cotton, or agriculture residue like sugarcane, bees case, and straw weeds. By removing non-cellulosic compounds like lignin, hemicellulose, waxes, and pectin, by which we can get pure cellulose compound, by which you can get pure cellulose fibers or powder in the powder form also, which is used for the paper manufacturing, textile, cotton, pharmaceuticals and biofuels. See, cellulose is technically a primary metabolite because it is essential for cell wall formation, essential for growth and survival because it found in all the living cells. However, modified form of cellulose may have some secondary roles for like defense or secretion, slimy layer, but usually it is not classified as secondary metabolites. And yeah, that is it for today. Thank you everyone for being till the end. Hope you like the lecture. And by this, we're done with this lecture. Soon we'll come with the next chapter. So stay tuned. Have a good day.